Good morning, welcome. So Carmen and I are here. Hopefully a few others join us, but I'm just going to go over a couple of pointers for cell organization. And as I do that, I want to show you Brother Deal on campus does his some notes for this way. And a lot of times when I see your notes, they're just pages full of words. I don't know about you, but pages full of words is overwhelming to me. So I would recommend that you find different ways, maybe try a few different ways. If you went through Pathway, maybe Cornell Notes, um, or maybe make some charts, or at least highlight the most important words so those stand out. So here, what he's doing is he took the cell in the middle and made this concept map. So here are all the organelles, and then using a different color for function and for structure, he outlined the structure and function of each one. And I'll keep working on maybe make one of these, we'll see. But you can see how this is going to help you organize the information. So you've got the Golgi apparatus and the function and the structure, and we'll go through each of these, but that might help you kind of process the information in a different way. Does that make sense, Carmen? Would that maybe be helpful?
Yeah, it does. It does help. Also, I noticed the intro video that you did in the announcements. That one, how you kind of linked it to like the FedEx and the trash guys and all that. That kind of helps a little bit more because it kind of gives me an idea of like kind of an example in real life to um, to remember them better.
Perfect. So you could even add in here like another color, a green. This would
Hmm.
be like the factory or whatnot.
Yeah.
And we'll
Yeah.
kind of point those out as we go through. So I'm just going to go through this PowerPoint and Carmen chime in. You're representing your classmates here.
Sure.
So the cell, if you notice behind me, it's Lego Mania. Legos are the building blocks that make up any kind of Lego structure. Cells are the building blocks that make up any kind of living structure. And they're pretty amazing. And understanding the cell will help you understand, of course, better how the body works. And of course, there are different cells and different parts of the body are going to be a little bit different, slightly different, but this is a generalized eukaryotic cell. And you can remember you are a eukaryotic cell as opposed to a prokaryotic cell, which would be a single-celled organism. So they're the basic living structural and functional units of the body. The study of cell is cytology. We'll just go through and hit on a couple of the main parts of the cell, their basic function. I'm going to go kind of quickly, and then I'll show you, I'm going to go through two of the quiz questions that people seem to be struggling with so far. So generalized cell, again. All right, so the plasma membrane is going to be the cell membrane. And we're going to spend all of module 5 going over the cell membrane. This is really important in how cells interact with each other and how a lot of physiology happens. So we're going to dig into that one deeply starting after 4, but when we get to 5. And then in the cytoplasm, which is the inside of the cell, we find the cytosol and the organelles. And then we also have the nucleus hanging out inside the cell. The cytosol is just that intercellular fluid inside of the cell, contains proteins, carbohydrates, lipids. We're going to just kind of cruise through this quickly. There's a lot of metabolic reactions that happen here. Then let's go into organelles. So if you think back to that first chapter when you had the prefixes and suffixes, if you go back and review those, it's going to make everything else easier. But organelles, I don't know that that one was on there. Organelles basically means little organs. So these are little structures inside the cell that have a specific function. The cytoskeleton is a network of proteins through the cytosol. It works to support. So if you're building that concept map, here are your functions to support the cell structure and shape. And it helps to organize those chemical reactions that are happening. It also helps in cell movement. And these are not something that are rigid. They are continually not necessarily rigid in shape and structure, but rigid in that they don't move, but they can be reorganized and moved. All right, centrosomes, these guys contain centrioles, and these are really important in cell division, mitosis and meiosis. Cilia and flagella, think movement typically with these guys. Flagella are going to be found in sperm cells, and they're going to help propel the sperm where it needs to go. All right, ribosomes. These are really important. The focus of this, as we go through the organelles, the focus really seems to be the process of taking DNA and transcribing it into RNA and then translating it into proteins. And the ribosomes are key here. They're the little spheres. They're made of RNA and proteins, and this is going to be they're going to be really important in this whole process of protein synthesis, or the making of proteins. They can be found on the endoplasmic reticulum, or they can be free floating. So you see here how they're made of a large subunit, a smaller unit, and this makes a complete ribosome that helps to make those proteins. If you look up ribosome on Wikipedia, there's actually a cool little visual, like a cartoon. You can do it in another tab if you want to, or note it for later, but it shows kind of how the ribosome processes. And I put it in a video on the collaboration board too. All right, so here you see the ribosomes attached to the endoplasmic reticulum, where they're going to be synthesizing proteins. This is a microscopic view. Kind of cool, huh? Oops, there was our endoplasmic reticulum. So the endoplasmic reticulum this is where it's continuous with the nuclear membrane. You can see how that it kind of just comes out, and these would, yeah, there's some space between them, but they kind of lay like pancakes and maybe not have lots of space between them, but they're going to have these ribosomes, and then the ribosomes are going to produce the proteins, and then the proteins are going to get released into this endoplasmic reticulum. And then here would be your function if you're building your concept map. Transport some substances, store newly synthesized molecules, synthesize and package molecules, detoxify chemicals, and release calcium. And this is actually a piece that is often missed, this calcium ion release. Clinical application. So one of the functions of the smoothie art is to detoxify drugs. Repeated exposure to drugs changes the smoothie R in the liver, which can result in the tolerance to different drugs. All right, Golgi apparatus. Again, we're going quickly. Like you said, Carmen, this is the in our analogy to a city, this would be like FedEx, or packaging and shipping. So here come, oh, go ahead.
No, I was just saying, okay, like, yeah, then that makes sense to kind of see a close up of it.
Yeah, so these, the endoplasmic reticulum is going to have these, part of the membrane is just going to break off into these little transport vesicles that you can see here. And you can see how this bilayer, or this, will just kind of pinch off and transport these proteins over here into the Golgi apparatus that will package and ship them to be taken out of the cell or to be used inside the cell. Kind of cool, huh?
Yeah, it's actually nice to hear about it than just to like read it and just see it. Makes more sense.
Yeah, good. I hope some of those little videos I posted too will help.
Hmm.
All right, so again, if you're going back to your concept map, here is the process, or the function of the Golgi apparatus: process, sort, and deliver proteins and lipids to the plasma membrane. or to lysosomes or secretory vesicles. All right, the lysosomes, these are going to be like the trash guys. Membrane enclosed little vesicles that contain powerful digestive enzymes. And their job there is to digest and take care of foreign substances. Interesting application here, Tay-Sachs disorder, which affects children of Eastern European descent, is caused by the absence of a single lysosomal enzyme. And if you look at the importance of DNA and genes and how one little change can cause a huge problem, this is a good example. That one missing enzyme that typically would break down a glycolipid commonly found in nerve cells. As a result of the inability to break that down, that glycolipid accumulates and nerve cells lose their functionality. So just a little snapshot of how important the function of each of these little organelles is. Peroxisomes, similar to lysosomes but smaller, also contain enzymes. And you can see right here part of their normal metabolic breakdown of amino acids and fatty acids. The cells are really good at recycling and reusing. This is part of that. They also oxidize toxic substances such as alcohol and formaldehyde. I'm going to shut my window because somebody's blowing in the yard next door. All right, proteasomes, structures that destroy unneeded, damaged, or faulty proteins. They contain protease. And this ASE ending is going to be typically an enzyme. So if you see ASE, think enzyme or something that breaks down. And they're going to break down, you can see the proteome of the proteins. All right, mitochondria. These guys are really important in synthesizing ATP. Hi, Rochelle, welcome.
Hey.
Feel free to comment. We're just kind of going through real quickly the organelles, and then I'm going to go over a couple of commonly missed questions so far.
Cool.
So the mitochondria have this double membrane. I think that's a quiz question too. The outer membrane is smooth, and then the inner membrane has these folds or cristae. These are going to increase the surface area for reactions that are going to produce ATP. And you could go into depth on this one. There's a lot to be learned here, but basically know that they've got these internal folds. They're just two layers. And how I like to remember it, when I was teaching this, I taught high school, and the cheesier we could get, the better, but we would be like, mighty mitochondria, and just remember that that's like the powerhouse of the cell that's producing ATP. Again, ATP production, and mitochondria are interesting. They have their own DNA, which is another cool topic. Let's see, mitochondrial DNA usually inherited from the mother. All right, the nucleus. So this is where I ought to rearrange this so that this is where we start, because the nucleus really is the most important, most prominent feature of the cell. I guess it's difficult to say the most important, but the nucleus contains genetic information that is vital to making proteins. Okay, try to remind him to make good choices. Sorry, my kids are on fall break, so trying to be entertained by Disney downstairs. So the nucleus, most body cells have a single nucleus. Some of the red blood cells don't, and then some skeletal muscles are going to have several. But typically they're going to have one nucleus. Parts of the nucleus include the nuclear envelope, which has channels or pores. We've got nucleoli, and then our big focus is going to be the genetic material. And you can think of the nucleus as kind of this protective place where the DNA can be to be protected. The DNA is packaged and tightly wound into chromosomes, and then it's protected here in the DNA. What would be the problem if anything were to happen to the DNA? To alter the structure or to damage even a small part? What would be the result?
Would that be almost like what you were saying right now, where one thing is wrong, then it can cause a big problem with something else?
Right, right. So the big idea in biology is that DNA is used to make proteins. And we'll go through the steps of that. But DNA is the recipe. So we can think of the nucleus as like the library. We've got 46 chromosomes, 23 from each parent, and each of those chromosomes would be like a bookshelf. And then each gene, which is a segment of DNA, would code for a specific protein or be the recipe for a specific protein. So it'd be like the book on the shelf that would tell us how to make that specific protein. All right, so the function of the nucleus, we kind of went through that. We've got 46 chromosomes. Let's see, you guys can read through that. They're going to package into chromosomes before they divide into meiosis or mitosis. All right, so again, here you see the DNA, the double helix. It winds around these little histones and packages into chromosomes. Chromosomes, you see the long molecules of DNA coiled together, and then kind of went through that already. All right, what I want to go over now is, I'm going to stop sharing for just a sec and pull up this other page. Hold on, sorry. Okay, hold on, sorry. All right, here we go. Buddy, I'll be right down, okay? Oh, okay. Thanks. All right, so this is the big key here, and I think this is what it seems like most of the questions, a lot of the questions were based around this process here. And we call this is Brother Deal from campus, and he's shared his lectures with me. He's pretty amazing. But I wanted to just share this screenshot of this one part. The central dogma of biology, and the big idea, if we could say like the central idea of the gospel, we would say is that Christ lives and the atonement is real and works in our lives, right? That would be the central idea or the main idea. If we look at the main idea or the central dogma of biology, it would be this process of making proteins from DNA. So we've got DNA housed inside the nucleus. Can you guys still hear me? Okay. Yeah?
Yes.
Okay, so we've got DNA housed inside the nucleus here that if unwinds and exposes a small segment called a gene that will be used to code for a very specific protein. So here's the process that we go through. This DNA is going to be read by RNA. And we're going to use the DNA, sorry, let me restate that. The DNA is going to be used as a template to create RNA. So then RNA, this process of going from DNA to RNA is called transcription. And DNA is built by nucleotides. Nucleotides are the building blocks that make up DNA. Nucleotides are also the building blocks that make up RNA. So ribonucleic acid, deoxyribonucleic acid, these are built by nucleic acids. They're the same language, so to say. So if we were going to take verbal language and write it, that would be the process of transcription, right? It's still the same language. So that's this converting DNA to RNA. And then we use the RNA, so those little ribosomes, they're going to run along messenger RNA. You're going to see how that process works. There's a couple of videos that I've posted, and then there's some in the course material too. You'll see how ribosomes run along that messenger RNA, and as they do, they grab little amino acids. And if you think of a polypeptide as a beaded necklace, and each of the amino acids that make up that polypeptide are the beads on that necklace, the ribosomes are what are going to add each of those beads and make that polypeptide. So here is translation, where we're going from the language of nucleotides to the language of proteins and amino acids. So building block here, nucleotides, building block here, amino acids. We're creating proteins, and we're going to go to a polypeptide, which is going to fold into that secondary, tertiary, and even quaternary structures to create our proteins. Does that make sense?
Yeah, it does.
Okay. So any
questions?
No, I'm good. I think I understand it this way.
Yeah, I think this section gives us a little bit of a breather. If you've had a little biology, it's not as hard. I want to show you now that just two of the questions that are most commonly missed so far. That's question 3. So when DNA determines the characteristic of the cell by specifying, and this is kind of basically what we just went over, the ultimate protein structure. Okay, and then the other question, question 18, and this again deals with that whole process of protein synthesis. Buddy, I'm almost finished, okay? The natives are getting restless here. So genes are responsible for the structure and function of all cells. And look at this closely. What was the result? We had DNA, we transcribed it into RNA, and then we translated it into, what did we translate it into?
Proteins?
Yeah. So this is the distractor here. Most people seem to be marking this one right here, but it is coding for proteins. So, all right, hopefully that helps. I'm gonna go make sure we don't have too much damage downstairs. All right.